welcome all. Nice full house here.
Yeah.
It's always fun introducing somebody who needs no introduction. So I'll maybe tell you a few things you didn't know about Jim. His first time here with the Kelp Forest Ecology course was in 1975, 43 years ago. Took it between undergraduate and graduate school. So Jim was a Stanford undergraduate, then went to Berkeley to do his graduate work. Worked with Wayne Souza there. Was Wayne Souza's first student. After graduating, but he did his most of his research down here in the kelp bed here. So he straddled those two rivals for his entire time there. Then after graduating, went to the aquarium and worked at the aquarium for 10 years before Chuck Baxter retired, and then we hired Jim to replace Chuck as our lecturer here. He's been here for 26 years and is going to retire at the end of the summer. So he has some long-term thoughts on what the ecology is like here in the kelp forest and here at Hopkins Marine Station. So turn it over to Jim to give us the long view on things. Jim.
Thanks, Mark. Full house today. Thanks, everybody, for coming. Hope I don't disappoint you. Anyway, so some lessons from the kelp forest is what I'm going to talk about today. And I've had, like Mark mentioned, I've had the great privilege of being able to park myself here on the shore of Monterey Bay for the last 40 years or so, and spend a lot of time out in the kelp forest looking around, poking around, looking at the natural history of the organisms that are out there. But since I've been here as a lecturer, lecturers at Stanford are not supposed to be doing research. Our main responsibility is to teach undergraduates. And so you have to sneak your research in on the side. And I've been really fortunate to be able to enslave my summer kelp forest ecology class to help me collect data. And so they get something out of it by learning the organisms and learning how they make a living. And I get some of the data that they collect. Some of you guys it might be too small to see, but that's Tony DiTomaso right there and Steve Wing right there, a number of years ago, though. So what I'd like to do today is relate some of the lessons the kelp forest has taught me over the last 40 years with the help of my summer students. And so the first one is rather a mundane one, but one I think we need to remind ourselves of fairly frequently, is that things don't stay the same out there for very long. They're not the same in the same place, and they don't stay the way they are at any given point in time. So as an example of that, these are abundances of the giant kelp, Macrocystis, with adult plants greater than 4 fronds. The red trace is what's gone on at Hopkins over the last 25 years or so. The blue trace is for Otter Point. And you'll note there's fairly good-sized standard error bars there indicating a lot of spatial variability. But even though these two sites are only 2 kilometers apart, when we do a scatter plot of just looking at the average abundances with their standard errors here, it's a shotgun blast. And there's no statistical correlation in the abundances of the kelp at each of those sites. And it's the same in a lot of ways for other abundant kinds of things. So here's 2 other abundant denizens of the kelp forest, at least formerly abundant. Pysastra giganteus, the predatory sea star, about whom we'll hear a lot later on today, and then the bat star, Pateria. And again, correlations between Hopkins and Otter Point here in numbers per 10 square meter. These are error bars of standard deviations there. But again, sort of a shotgun blast. The correlations actually are significantly different from 0 here, but huge amount of scatter. And note the difference in the scale on the Otter Point axis compared to Hopkins here. And then just lots of variation for the bat stars. So that's for abundant things. So things you can actually go out and, in a reasonable amount of effort, get some numbers on. We ecologists love to count things, so there's a lot of count data that we'll see today. So for the abundant things, you can get these kind of patterns, but you also find out that knowing what's going on in one place doesn't really tell you a whole lot necessarily about what's going on in other places. But another one of the lessons that I've learned is that most species are rare. And some of the rare species sometimes surprise you and show up in unexpectedly large numbers. And that's happened several times during the course of my stay here. And one of the first ones that happened was the little tube snail here. It's called Serpulorbis squamigris. It's actually a snail, even though it looks like a tube worm. And it sits in one place its whole life, spins a nice little mucus net that captures tiny phytoplankton that it feeds on. And when I first started diving here, you could find it. It was spotty here and there on the substrate. And then in the early 1980s, sort of without really noticing when it happened, all of a sudden you saw these guys everywhere. And especially in the rocky intertidal, we saw densities in the mid-intertidal zone with these guys just cheek and jowl like that here, sort of like what you see in Southern California, but hadn't really been occurring here in Monterey. And then over the years, this cohort sort of got slimmed down through attrition, but there's still fair numbers of them out there, and probably having intermittent kinds of recruitment because the larvae have gregarious settlement and they settle where the adults are. So that was one of the first of these unusual sort of big pulses of things that happened. Another one, and this will be a player that we'll come back to at several points during my talk today, is the little subtidal acorn barnacle, Balanus crenatus. It's the cousin of the intertidal Balanus glandula that probably more of you are familiar with. It's a smaller barnacle, tiny little guy, and during the times when I was first getting acquainted with this place, we would see it, but it would be in mostly disturbed places, particularly small patches, like where a big kelp holdfast gets ripped up, creates open space, and the barnacles would be one of the first guys to get in there and colonize. And then also on large vertical rock faces where the layering of the cover is a bit less and there's usually a little bit more open space, those would also be places we'd see patches of these guys. But then in two periods, in the early 2000s and then again starting in 2010, these guys were just everywhere. It was raining barnacle larvae out of the water column, and they were almost 100% cover on everything. They'd settle on the blades of the algae. They were even settling on the big gumboot chitons and other sort of soft-bodied things. They were everywhere. And they would settle really densely and then grow up like barnacles are wont to do when they're really densely packed into these tall columnar shapes because they're really tightly packed in there and limited for space. And then they break off easily, leaving these big patches. And this is sort of a substrate level view that, say, a little larva trying to come in and recruit someplace would see on the bottom during these times. Is just wall-to-wall barnacle legs up there feeding, grabbing larvae out of the water. So if you're a larva trying to settle and gain some space, this period, these two periods were just like barnacle hell out there. It was just wall-to-wall barnacles, and if you were trying to get to the substrate, you'd like as not probably get eaten by one of these guys. So, unusually large settlements of these guys. Now, of course, like a lot of things I'll talk about today, I wasn't making my students collect data on these guys. We weren't counting barnacles because they're patchy and sort of hard to get accurate measures on. But looking back through the data that I have had the class collect, one of the things we've been doing is looking at the diets of Pisaster giganteus. And so here what we see is a frequency in the diet. So we just go turn starfish over, look to see whether they're feeding, and if they are feeding, what are they eating? And so this is the fraction of the feeding stars across different years that happen to be feeding on Balanus crenatus. And so I'll let you guess which years there was lots of Balanus out in the kelp forest there, because Pisaster is a really opportunistic forager. We'll see some more of this, some more of their diet data a little later on in the talk. But at least by asking the Pisaster, what are you eating, that tells us something about the abundance of these really common and easily consumed prey like the Balanus. So, early 2000s, big spike in the occurrence in the diet, corresponding to big spike in the environment. Likewise, starting about 2010 and then going to 2014 here. So, we see these big spikes and changes in abundance of species that come at unexpected times and at unexpected intervals. And probably the champion one of this is the sea urchins, both Strongylocentrotus purpuratus and now Mesocentrotus franciscanus, the big red urchin. So, timescale along here, these are data from John Pierce's and Tuck Hines' sort of classic paper on urchin population dynamics here, right here at Hopkins. And during the 10 years that they were sampling with their students, there was 1 year where there was a big spike in the purple urchins, and then a gradual decline, or a fairly quick decline, back to baseline levels here, well less than 1 per square meter. And then for the next 30 years, that's what persisted. Just a fraction of an urchin per square meter. Present, but hardly worth noticing. These data are data that Theo and her students collected when Theo first got here. And then I said the kelp forest class would take it over, and we did it for a few years, and there were so few barnacles, there were other things we wanted to do, so we just sort of, or so few urchins, that we just sort of stopped counting them because they had been in such low abundance for so long. And then, of course, as soon as we do that, they show up. And so again, we've got this span of 30 years here with hardly any sea urchins at all, and then a big spike. And this appears right here off Hopkins to continue to be going up. And so we'll come back to these guys a little bit later on in the talk, but this is just an example again of this really intermittent pattern of recruitment, where if you're just looking at these populations over the typical length of a PhD thesis or the typical length of an NSF grant, like as not, you'd never see one of these events. But as we'll see, they can have really important consequences for the entire community that's out there. All right, so that's one lesson. Most species are rare, and then every now and then we get these unusual events of things all of a sudden showing up sort of out of the blue. The other lesson, the next lesson that the kelp forest has taught me, is that history, past history, and the ecological context in which different events happen make a big difference in both our interpretation of what the interactions are that drive these things and what the potential actual consequences for the communities might be. So this one's going to take a few minutes to develop, a few slides to develop here to tell this story. But it comes mainly from photographic plots that I initiated in August of 2011. And again, this was by enslaving my summer class. I made them go out and we drilled a bunch of holes in the granite to put permanent markers in for these half-square-meter quadrats. And we've got 52 of them located in 3 sites around the kelp bed. This is Hopkins right here. So we have some out on the west side, some right off, straight off Bird Rock here, and then in the near the 100-meter buoy site on the east side. And these are on vertical rock walls, mainly because the vertical rock walls have less layering of things, and so it's easier to see how the quadrats are changing on these vertical surfaces, because the horizontal area is more dominated by algae, and they get tall and waft back and forth, and it's hard to see what's underneath them just within the photographs. So they're half-square-meter photographs. Here's a picture of the framer, size of the framer, and photograph them 3 times a year. In March, right at the end of the winter, at the beginning of the upwelling season, but prior to the big spring-summer recruitment that a lot of the inverts, the pattern that a lot of the inverts show here. And then in August, at the more or less end of the upwelling season and after the spring-summer recruitment. And then in December, right before the big winter storms usually come in, and after the oceanic period. So 3 times a year since 2011. So what I'll show you here is a sequence of photos from one of the quadrats. And this is one, I believe, that's out on the west side here. So in August 2011, this is what it looked like. This is Corynactis, the strawberry anemone here, Dideminum, a little compound tunicate, and then all this sort of whitish stuff here and over here, that's all Balanus cornatus. That's the acorn barnacle. Because this is 2011, the big barnacle settlement started in 2010. So, summer, end of the summer, lots of barnacles. December 2011, still lots of barnacles, but some holes and patches in the abundance of them. And then by March, hardly any barnacles left at all. So there's a few barnacles left over here, but some crustose corallins showing, some foliose red algae starting to show up here and take over some of the space. So in a 6-month period, most of the barnacles that were just dominating these plots in August had disappeared. And then in August, barnacles had settled again, grown in, covered a lot of the space. Again, on both sides of the big strawberry anemone patch. And then by December, again, sort of thinned out, some barnacles still present, but much thinner. By March, encrusting bryozoans, encrusting corallines, very few barnacles present. And then August 2013 again, another big settlement, huge cover of barnacles. And so that just kept going on for several years. And so what we see here is what I did was I took at least 10 quadrats from each of the 3 sites and just did a quick eyeball qualitative estimate on barnacle cover and ranked them from 0 to 5, or less than 5% or greater than 85% cover. And then what I plotted for each site is the median barnacle cover from these scores here. And you can see that all 3 sites, even though they weren't doing exactly the same thing, the general pattern is the same. Highest in late summer, dropping through late fall, and lowest levels early in the year in March, and then going right back up in August. So this indicates that during these years, rather than just seeing barnacles that persisted because they're long-lived, we were seeing this really unexpectedly fast fluctuation in the barnacle population, up and down, until the end of 2014, and then it's pretty much just shut off. You look at the photographs after this, and there's very little barnacle cover in them. And the next picture here, this is from December 2017. And so this is that area that was just dominated by barnacles here and over here. And now it's covered with probably five species of erect bryozoans and a couple of species of encrusting ones, some crustose corallines still showing through, big compound tunicates, which turn out to be extremely dynamic in these images. And some of this white stuff here that might, at this resolution, look like barnacle cover is actually serpulid polychaete. They're calcium carbonate tube worms. So hardly any barnacles at all in here in December 2017. So once the barnacles stopped settling, the diversity, benthic diversity of these plots just went way up. All right, so you might wonder, okay, what was driving this other than the strong annual recruitment of the barnacle? What was driving the loss of the barnacles? And for those of you who've been out there and know something about the organisms that live there, of course, the culprit was the sea star Pisaster giganteus. So the procedure, which I didn't tell you about a few minutes ago, the procedure when we go out and shoot these things is to put the quadrat in its place using the marker pins, take the picture, and then rip off all the big stuff like sea stars and cryptochitin and other things that are covering all the small sessile stuff. And so this is one of the plots with, at that time, a fairly typical number of stars when there are lots of barnacles around for them to eat. Because barnacles, if you're a starfish, are pretty much the easiest thing you can eat. You sit there, stick your stomach out against them, and you don't even have to move to digest them, and then you just walk a few centimeters to one side or the other, and there's a whole other meal right there. So they stay put, they feed at high frequency, and can gain a lot of calories from just eating these sessile barnacles. All right, so another thing that happened during this period, of course, that most of you remember is right here was when we had the big sea star mortality event. And this, at least right here off Hopkins, resulted in loss of 75 to 80% of the sea stars in terms of numbers here. So middle to late summer 2013 is when it started and continued through the fall. But what's going on here? If we lost 80% of the sea stars right about this point in time, but yet the barnacle abundances continued to drop, and then the following year's recruitment that generated a spike also dropped, but only in the presence of about 20% or so of the population of sea stars that had been there previously. So what was going on with that? Well, another thing that comes out of poking your head around in these places for long enough is you learn who some of the other players are. And one of the important players here is this gastropod, ceratostoma, called the leafy hornmouth because it's got a big spike on one side of its aperture here. And it's a barnacle predator. It doesn't eat only barnacles, but when it can get barnacles, that's probably its preferred food. But for the purposes of our story here, the way it reproduces is the most important thing. What these guys do is they get together and copulate and lay benthic egg cases, and then they go through their entire larval phase within those egg cases and come out of the egg cases as little crawling miniature juvenile snails. And a lot of the neogastropods tend to reproduce that way, but most of them tend to have planktonic larvae that go off and swim in the plankton for a number of weeks at least. But these guys have crawl-away juveniles, and so that allows them, when there's these big bonanzas of food like we saw between 2010 and 2014, to respond at the population level very quickly to changes in resources. And so here is one of the quadrats, and you might say, well, what is that? And you got to look real closely, but each one of these little lumpy things in the photograph is an individual ceratostoma snail, and this is a corner of one of the photographs. So in about a quarter of one of those half square meter plots, it's covered with snails. There's about 30 or 32 of them in here, and they're getting together and fornicating wildly and laying egg cases that at some instances cover a good proportion of some of the photo quadrats too. And it's during this period that it's really the only time that I've ever seen little juvenile ceratostoma. And this guy is maybe a centimeter long, really cute little snails. But up to this point, I'd never seen a small one like that. And again, these guys are produced by crawl-away young, little miniature baby snails that come out of these egg cases. And then they immediately, when there's this many barnacles around, they can hardly miss them. And so they start eating barnacles right away. And the other surprising thing is, evidently, they must grow fairly quickly, too, because the abundance of adult snails went up really quickly. So, of course, this was another thing that we weren't actually counting and keeping tabs on. And so the only data that I can get is by looking at the photoquadrats and counting the numbers of ceratostoma that show up in the images. And one of the difficulties with that is that Pisaster eats these guys too, as well as barnacles. And so the snail has a pretty well-developed escape and avoidance response when the Pisaster are nearby. So if there's lots of Pisaster in the quadrats during the time before the mortality event, it's likely that many of the ceratostoma that were around were probably not right in that neighborhood because they'd vacated because of the high number of Pisaster there. But you can still see that there was respectable numbers of snails still that even showed up in the quadrats here. And my suspicion is that the high abundance of prey starting in 2010 led to high reproductive rates of the existing ceratostoma, and because that barnacle settlement persisted for several years, it allowed the population to respond. And because of their benthic embryonic development, allowed their population to increase a great deal. And once the Pisaster died, this had gone on for enough years that there were enough ceratostoma to continue along with the remaining Pisaster to drive that annual cycle in the barnacle population. And you can see that in the quadrats after the sea stars disappeared, that we started seeing a lot higher numbers of predatory snails in these quadrats, particularly in the years where there were still some barnacles around. So, finally, get to the point here about why history matters, is that if you think about, well, what would this have looked like if the Pisaster mortality had occurred at a different time instead of the time that it did? So, for instance, if the sea star mortality had happened right before 2010, before the barnacles started settling really strongly, what probably would have happened is the first cohort of barnacles would have settled, but because the Pisaster weren't there to eat them and the ceratostoma hadn't had enough food for a period of years to augment their population, that barnacle cover, instead of showing this cycling, probably would have persisted and even gone up to a much higher level, being annually reinforced by subsequent recruitment. And we would have seen barnacle domination possibly of these habitats for a very long time. And people would probably have also tried to infer that loss of the pisaster led directly to recruitment and survival of the barnacles and domination of the habitat, even though it was probably due to larger scale conditions that just changed the transport and delivery of larvae to these habitats, regardless of the presence of a predator like pisaster. Another thing, if the mortality had occurred at the end of this period of barnacle settlement, we probably might not have seen much difference in what subsequent changes in these quadrats look like, because the barnacles would have stopped settling. And then lastly, if it occurred where it did, but we hadn't been keeping track of other stuff that's going on or knew about the natural history of some of the other alternative predators to barnacles around here, we might have even decided that because this cycle in the abundance of the barnacles up and down continued after the pisaster died, that pisaster wasn't playing that big a role in these fluctuations beforehand. And so we would have gotten a different view of what the role of this sea star was playing, simply because of the context in which this particular mortality event happened to occur. So instances like this have convinced me that history is really important when we're trying to study ecology and looking at the consequences of changes to various aspects of these communities. You have to know what else is going on with the other players and what has gone on in the past. All right, next lesson is really the simple abundance of a species doesn't really tell us exactly what it's doing. Okay? And there's a lot of programs that we have, like the PISCO sampling program and the Reef Watch program, where people go out and they do a lot of counting and censusing of organisms, because we ecologists, again, love to count things. And those data are used as sort of benchmarks for the health of these communities and what's going on with them. And while I applaud that, because we need the more people we have out there looking at these communities and keeping track of what's going on, just knowing how many things are there doesn't necessarily tell us the whole story about how these communities work. So, since we're talking about starfish, let's continue with starfish. So we'll look at abundance of pisaster, and this is again data from my summer class, where we go out and we drop randomly placed quadrats, circular plots, 10 square meters in area, count all the sea stars in them, turn them over, and see what they're eating. And then these are the mean densities with standard error bars, plus or minus 1 standard error. And you can see from year to year, it fluctuates up and down. Some of this stuff may be real, some of it may be just sampling error. You can see there's large error bars on here, which suggests that these guys are really patchily distributed. This wasn't a posed photograph. This was a clump of pisaster preying on a clump of barnacles here, so they're in really high density. And you get some of these clumps in a quadrat. You get high values for that one, but other quadrats would have very low numbers. So if you squint your eyes, probably the overall long-term average is something just under 2 stars per 10 square meters, which is fairly abundant for a good-sized predatory animal out in your habitat. Of course, 2013, this is where the mortality event occurred, and since then, pisaster has been considerably lower in abundance, a lot smaller standard errors, so there's consistently very few of them out there. And when this happened, I thought, oh, this is going to be really interesting, because we've got this big, massive die-off and loss of the most abundant benthic predator out there, and so we're going to really see big effects, and we're really going to be able to see what role this guy is playing in how the benthic community dynamics works. But instead, not much has happened. There hasn't been anything really, really obvious that's gone on. And I was sitting there scratching my head thinking about that. Why haven't we seen these big effects of this dominant, large, active predator out there? And when I started really looking at the diet data that I make the class collect, so these are diet data here. This is one of those slides I always tell my students, never make a slide that looks like this, because there's too much stuff on it. But that's actually the point here. So we go out every year, first of all, I make the students learn all the organisms so that when they go and turn these things over and see what they're eating, there's actually some chance they can accurately identify what the prey is. And they gladly do this. It's really gratifying to see them that interested and enthusiastic about doing all this work for me. So these are the diets. These are years along here. The first numbers in each column are the number of sea stars that were sampled in that site in that year. And then the proportion here is the fraction that are feeding. And the data from Otter Point out here and right off Hopkins here. Huge diversity in the diet. So almost 80 species. About 30 of those only occurred once during the whole period that we've been monitoring the data, so you might be a little suspicious of those. But even so, that leaves a diet of 50 species in both of these sites. So Pisaster, first of all, eats a whole bunch of different things. And if you look here, I have listed the two most frequent prey each year and each site here. And the variety of things they eat is just astounding. So Serpulorbis and Petaloconchus are these little tube snails, the sessile vermidid gastropods. Balanus crenatus, of course, is here, the barnacle. But a variety of other sessile kinds of things. Phragmatopoma and Sabellaria are polychaete tube-dwelling worms. Lots of mobile gastropods, Tegula, Calliostoma, Ceratostoma, Pomalax. All of these guys are mobile shelled gastropods. So these guys are eating a whole wide variety of different things. And if you do a sort of a quick and dirty cluster analysis on this, so this is similarity along this axis, and then the blue years are data from Otter Point, the red years are from Hopkins. And the only time when we see really high similarity amongst pairs of years or sites is during the barnacle years, when pretty much everybody was eating barnacles. But even in those years, the total number of prey that we observed was still fairly high and not that much different from the prey diversity or prey species richness in the non-barnacle years. But in the non-barnacle years, even the most similar years only have about 60 as a score for their similarity. Some of them are much different than that. And where you see clusters of years of the same color, you never see or rarely see here consecutive years that are the ones that are most similar to each other, which suggests from year to year and place to place, the diets of Pisaster are really, really different. And so this is one of the things that makes me think this is why Pisaster hasn't had a really hugely obvious effect when we saw the mortality event, because it interacts with a lot of species, but with any single species, that interaction is very weak. Okay, so it's a very diffuse kind of effect across a wide array of species, each of which is fluctuating and changing from year to year on its own sort of schedule. And Pisaster is just eating what's either most easily caught or things that are most abundant. Now, the stuff that we saw in the photographs and the photoquadrats clearly shows Balanus can actually change the abundance of its prey. But Balanus is sort of special because it's sessile. The stars don't have to put out any effort to capture it at all, and it can just sit there and digest it. Whereas a lot of these other prey, especially the mobile ones, it's got to chase them down and exert some energy to try and catch them. Also, there's a possibility, one of the other reasons why we haven't seen big effects of Pisaster is a lot of the things that it eats, particularly the gastropods, typically are long-lived and they have very intermittent recruitment, so maybe we just haven't seen the consequences of the loss of the Pisaster yet. But still, I think the diet data show us that just knowing how many sea stars are there really doesn't give us at all a good feel for what the role it's actually playing as a predator in these communities. Now, another instance, and this one's probably a little more dramatic, has to do with sea urchins in terms of just knowing whether a species is present or absent or how abundant it is, telling us what's going on with the community. So again, this is the time series with a 30-year hiatus of really low urchin abundances here in the Hopkins Marine Life Refuge. And of course, right here, 2013 is when the sea star the sea star mortality occurred. And of course, when you see a pattern on either side of a big event like that with low abundance of something on one side and high abundance on the other, everybody's first thought is, there's a cause-effect relationship here. And that loss of the sea stars allowed the sea urchins to increase. And I don't think that's correct for a variety of different reasons, all of which admittedly are circumstantial. So first of all, early 2014 was when in the photo quadrats is the first time that a sea urchin shows up in one of the quadrats. And by then, it is 20 to 30 millimeters in test diameter, and an urchin of that size is probably at least a couple years old already, suggesting that if we saw it in 2014, that it was actually settled back in 2012 before the mortality of the sea stars. Also remember, these quadrats, the photo quadrats, are on vertical surfaces, which are not the urchin's primary preferred habitat. And so for them to show up in these vertical places suggests that there's probably lots more urchins around in places other than the ones we were photographing. Then in 2015, January 2015, Giacomo Bernardi up at UC Santa Cruz, he and Nicole Crane were out diving at Point Pinos, and he sent an email to John Pierce, which John forwarded to me. And Giacomo says, we were out diving at Point Pinos, and it's an urchin barrens. There's these huge sea urchins out there. And Giacomo said in his email that he saw purpuratus that he didn't know they got that big. And they must have been, from measurements we did subsequently, they must have been 60 to 80 millimeters test diameter. Big, huge guys, which have got to be at least 6, 8, maybe older years old by then. And then, looking at my field notes and my dive log from 2011 from diving in northern Carmel Bay, an offshore set of reefs called the Pinnacles, in my dive log I have some notes that there are all these little urchins at that time, around 20 millimeters, 20, 25 millimeters, in clusters, creating small grazing halos around small shelter areas and crevices where they were. So small urchins, high abundances, in 2011, but my field notes from 2010 didn't record that we saw anything like that in 2010. And the guys that were of the size we saw in 2011 are probably about a year old by then. So for all these circumstantial reasons, and this photograph that you see here with this high abundance and cluster of urchins was taken in 2008 right out here at Hopkins. So for all of these reasons, my thought is that the urchins were already present, and maybe small in size, but already present and settled at the time that the big sea star mortality occurred. And if we just look at the structure of the population, this size frequency data just of purpuratus, the purple sea urchins, in 2015, 16, and 17 right here at Hopkins, and then 2016 and 17 out at Point Pinos. And you can see there's fairly big urchins out here. I mean, these guys are gargantuan ones for what we normally see. And even bigger and higher densities out at Point Pinos than at Hopkins. And this high abundance out at Point Pinos has really created a deforested area. So what is typically in the literature called an urchin barrens, with lots of sea urchins and mostly crustose corallins and a few hardy cnidarians. Started out here at Point Pinos in late 2014. By 2015, the summer of 2015, it was here off Coral Street. Otter Point was pretty much deforested by 2016, and then last summer it came right up to the middle of the kelp bed here at Hopkins with just little stands of giant kelp hanging on surprisingly in the nearshore environment here along the whole Pacific Grove shoreline. And otherwise, it's just a big urchin deforestation out there. And so, of course, the thing that probably comes to mind is, what have the otters been doing out there? Because they're the 400-pound gorilla, or I should say the 40-kilogram mustelid, sitting in a room here, and everybody knows the sea urchin, or the sea otter sea urchin kelp story, right? The sea urchins eat the kelp and have a negative effect on the abundance of the kelp, but because the sea otters are present, they eat the sea urchins, have a negative effect on them, and therefore indirectly have a positive effect on kelp. And the reason that we have kelp, so the generalization goes, is because of the otters keeping control constantly of the grazer population. And this is just, it's embedded in our psyche almost, that the otter is sort of the guardian of the kelp forest. So why the heck should we get these deforestation events right in the middle of sea otter country when abundances of otters, at least in southern Monterey Bay, have stayed constant or even increased a little bit, even though distribution-wide up the California coast, there's been some loss of otters or reduction in their population over the last few years. It's really not been real evident here in Monterey. Certainly not to the extent that it would have to be before you would expect from the otter urchin kelp scenario to expect deforestations to occur, but yet it did. So what the heck's going on with that? Well, thinking about this prompted me to remember all the things that we actually already know about how this interaction works and how complex it is. And one of the really important things, of course, is that otters don't eat just any old urchin. They ignore things that are small. And so anything less than about 20 or 30 millimeters, they just don't bother with. In all the time that we've been monitoring the diet of piesaster, 1,500, 1,600 observations of feeding, we have never actually caught a piesaster in the act of eating a sea urchin, even a small one. And they probably do once in a while, we just haven't caught them doing it. And then the 20-armed sea star, Pycnopodia, it is an urchin predator, but here along the inner Monterey Bay side of the Pacific Grove shoreline, Pycnopodia has never been all that abundant, and probably not abundant enough to actually control the urchin populations even before the mortality event. So one of my thoughts is that the absence of large numbers of sea urchins for that 30-year span between the late 70s and the about 2010 was due to limitations in recruitment and supply of new individuals to the population rather than necessarily predation. I mean, otters are probably eating some urchins, but there wasn't enough of them around for them to really concentrate on them. And then there's the social behavior of the otters. Otters, as a species, have a very broad diet. They eat a bunch of different things in order to fuel the fires that they need to keep themselves warm. But each individual tends to specialize in a subset of that broad diet. And the females tend to train the pups to those same diets. So if they're specialized on, say, turban snails, the pups are specialized on turban snails. If they eat a lot of mussels, the pups learn that mussels are good to eat, and so they eat those things. And so they pass these things on through the social structure of the sea otters. They pass their food preferences on, or food selectivity on. And so it brought up to mind whether or not the persistent low number of urchins across 30 years, which is probably 3 generations of sea otters, did that lead to a cultural memory loss that you can actually eat these urchins? Because there wasn't enough of them around for anybody to specialize on and to teach their pups that you can eat these things. And so every now and then one of them might pick up an urchin that comes out of its crevice and is available. But then when a whole bunch of them show up, it's like, what are those spiky things down on the bottom? And so you might think, well, yeah, that's a good story, but it sounds sort of like BS to me. But there is a precedent for this. And this is another thing that happened during the last 40 years or so. And this is the big whelk, Coletia. It's a big snail, gets about this size, lives down in Southern California, and started showing up in large numbers here in Monterey in the late 70s. And through the 70s to the mid-90s, it just kept getting more and more and more abundant. And you would come across places in the summer where there is areas 5, 6, 8 meters on a side that were just wall-to-wall Coletia mating, laying eggs, reproducing. And I always thought, how come the otters aren't eating these guys? They're these big, meaty snails. Other things eat them, so it didn't seem like they were noxious or chemically protected. But why weren't the otters eating these guys? And it got to the point where abundances were about 0.1 to 0.3 per square meter, so you could go out there with your eyes closed and put your hand down on the substrate, and there'd be one of these guys within a meter of you. So they were really abundant. And then between 95 and 96, all of a sudden, their abundance dropped by an order of magnitude, and there were busted shells everywhere out in the kelp bed, which could have only been done probably by sea otters, fracturing the shells, because they're big, heavy, massive sorts of shells. And so the otters did finally figure it out, but it took them 15 years, a time lag. And again, I think possibly that's due to this sort of cultural passing on of food preferences and what's decent to eat and what's not. And it takes some Einstein otter to go down and sample one of these guys, a real foodie otter to go down and sample one of these guys, bust it open and say, wow, these are good. And then the other otters are watching it and say, oh, what's Fred eating over there? You can eat those things? And then they just clean them out. And they did. So Colletia is present out here in the kelp bed still, but it's not nearly as abundant as it was before the otters figured out that they could eat it. So this is the thing that gave me the suspicion that maybe the reason that the otters are not eating urchins as much right now is that they're still in the process of rediscovering that these things are good to eat, simply because they have been so rare due to lack of recruitment for a long period of time. And so to me, the really important thing of the occurrence of this deforestation is it sort of really made me reevaluate my assumptions about how these places work. And just subconsciously, I've always thought, oh, we don't have urchins out here because there's otters. And while that is indeed part of the equation, there's a whole bunch of other stuff going on in here. And Mike Foster and Dave Scheel in a paper back in 1988 aptly asked the question, are sea otters keystone species, or are they just another brick in the wall? And the occurrence of this deforestation here in Monterey, smack dab in the middle of sea otter country, has sort of made me think along those lines too, but rather than a static wall made of bricks and parts that create these communities, I think a more apt analogy is that these things are like gears that are intermeshing, but the size of the gear grows and shrinks depending on the biological conditions, so that sometimes those gears mesh and influence and turn other things, other times they shrink and the teeth don't mesh and they don't interact as much. And there's this constant shifting and changing of all those interlaced gears that drive the dynamics of the communities that we see. And otters are a big part of that whole assemblage, but it's not the only thing that's going on. So to me, the most important lesson I've learned from this event is that this otter urchin kelp scenario isn't wrong, it's just massively incomplete. And if we let our subconscious assumption about what's important and what's driving the community changes that we see out here become dominated by these sort of simple little 15-second sound bites, because those are the stories that we can communicate easily and quickly on an elevator ride, then we lose sight of the true complexity of how these communities are put together and how they work. So a corollary of this lesson here is that it's really important not to think of species as just black boxes, little automatons that actually, whenever they're present, they do exactly the same thing in exactly the same way every time they're there, because they don't. Otters forget that you can eat urchins because they didn't learn it from their mom, because their mom didn't have them to eat. And so knowing something about the details of the biology always makes our lives a little harder as ecologists, but it adds that necessary complexity to our understanding of how these places work, because that's how biology works. So it'd be nice if we could have nice, simple stories that we could tell and that were accurate and deterministic and predictable every time a given assemblage of species occurs. But biology is more complicated than that, which is what makes it much more interesting. Okay, I've been yammering on for a long time here, and so I put this slide in just to remind myself to tell you that it ain't just been about barnacles, sea stars, and sea urchins over the last 40 years. Every one of these little mug shots up here has a story with it. A story of unusual change or times that you couldn't find that particular species and then it shows up again. And so there's a lot of untold stories, albeit ones that I probably don't have as good data on as some of the stories I have told you. But the things that I told you today are not exceptions. They're just exemplars of a whole host of similar sorts of dynamics. Change and constancy and unexpectedness. Now, the last lesson that I've learned, again, being an ecologist, I love to count stuff and play with statistics and do all that quantitative kind of thing. And what playing around in the kelp forest for 40 years have told me is that you can't ever have enough data. And anytime you have data, it ain't going to be enough to answer the questions that you have. First of all, because you can't count stuff that isn't there yet. And sampling programs that you try and get going, you can't look into the future and see who's going to be there, so you should count it today so you can see how it's going to change when it does get really abundant. Also, because of the variability that we see, it's really hard, if not impossible, to get any true replication in time, because the context in which the communities exist, whenever we ask our questions in an experimental way and do a manipulation, the context is going to change depending on the time when we do that. And unfortunately, we ecologists never repeat our experiments. If we get one to work, we never repeat it. And I think that's crucial that we do that because of the changing background. And an experiment may occur one way at one time because of the assemblage of species that are there, but the next time we do it, the assemblage is going to be different, and the result of that experiment, that manipulation, may be completely different. So it's really hard to do experiments, especially if you don't have controls, and of course natural experiments, everybody knows that they don't have controls, and so they're not really true experiments. But against this background of change, it's really hard to figure out how do we best study these kinds of communities. The other thing, too, has to do with sort of our perceptions. And a lot of this came out of discussions I had with John Pierce and Chuck Baxter about how things changed over the long term. And this was in the context mostly of intertidal studies, but I think it applies even better to subtidal areas. And those kinds of discussions have led to a really, really interesting historical ecology project that John and Theo are primarily spearheading, in which we're talking to a bunch of old-timers and trying to interview them about what things were like when they were here at Hopkins, either as graduate students or some other capacity where they looked at these communities. And reading through the transcripts of those interviews sort of prompted my production of this little figure here. So we have 40 ticks of the annual clock along here, and my imaginary but not unrealistic trajectories of a bunch of different species through time. And we can't help, when we first encounter and start learning the details of a particular biological assemblage, we can't help but put that time as our baseline. That's, regardless of whether it really is or not, that's the pristine condition. Those are the good old days. And anytime we come back and look at those assemblages from some distant point in time, we compare them to what it was when we first saw it. And depending on where our baseline is along a set of trajectories like this, each of us might have a very, very different view of what that pristine condition is. And if it's different when we come back and look at it, then we might think, man, things have gone to hell. It's really different than I remember it. But depending on where we are in these cycles, one possibility is that we're just seeing a different part of these whole big trajectories that change through time. And so the more, from discussions with Chuck and other folks, but mainly with Chuck, the more I learn about cognition and how our brains work, the more suspicious I am about what I actually remember and how true it is, because our perceptions and what we think we remember changes through time. And we can't help that, but it's just the way it is. But also the world changes around us. And so, to me, one of the really important roles of a place like Hopkins and a place like Marine Labs is to provide that sort of institutional level memory about these kinds of things. That it's not only an opportunity we have being parked here on the seashore, but it's an obligation that places like Marine Labs have to keep a finger on the pulse of what's going on, to monitor and provide institutional memory that's hopefully a little bit more quantitative and a little bit more accurate about what these fluctuations are looking like, and not have to depend on the failing memories of all us old guys who may not remember it exactly the way it really was. And so that's really emphasized to me the importance of following places for a long period of time and taking advantages of our location here with all this stuff going on right out our back door. It's really inexcusable not to keep a finger on the pulse of what's happening there. All right, so final slide here. So these are really sort of lessons that I've learned that I wish I could go back and tell myself, back when I was a snotty little arrogant know-it-all undergraduate who first showed up here at Hopkins. If I could go back and tell that person some things to sort of look out for, this might be at least a short list of some of those things. And first of all, it would be to watch places for a long time and keep better notes, way better notes than I did. And then secondly, get students to help while they learn. And I think I've been able to do 2 out of the 3 of these here. Is I've parked my carcass here on the shore of Monterey Bay for a long time, and I've coerced my students into helping keep a finger on that pulse, giving them the tools, teaching them the organisms and their biology and generating their interest in it, and then employing what they know to help keep a finger on that pulse. Also, being sort of a data nerd that I am, we got to do experiments. We have to do manipulative experiments. That's the only way we can, when they're feasible, that's the only way we can figure out what's actually going on. So do them when they're possible, but don't expect them to replicate if we repeated them at different points in time. And actually, one of the things we should do is repeat them in time so we can figure out why we don't get the same result. And that leads to better understanding of how these communities work. And again, I say this being a data nerd and sort of, teaching statistics and playing with statistics and being very interested in it, but I think we should worry less about p being less than 0.05 in statistical significance, worry less about our generalized linear mixed models and our big meta-analyses with sort of Bayesian likelihoods attached to them, and pay more attention to the biology that's going on. Learn about the idiosyncrasies and the specific aspects of the biology that goes on right outside our back door, so we can interpret the changes that we see, both in a quantitative sense, but also in a biological sense. Second to last lesson is don't treat these unexpected events that occur as nuisances. It's not noise that occurs that we need to average out, but it's part of the fabric, the tapestry that forms these communities and drives the dynamics oftentimes for long periods after these sorts of events occur. And so they're not noise. They're not junk that we need to average out. They're things that drive the system, and we need to understand them better. And then lastly, need to be really beware of thinking in short, straight lines. Because our short attention span world that we have today, the fact that even a PhD thesis only goes for 3 years, or an NSF grant only goes for 3 years. And during that time, you got to pump out the publications so you can get your next grant. And it forces us to think in short chunks of time. And that's not necessarily how ecology works. And so these simple stories that we can come up with aren't necessarily wrong, but they're probably almost always incomplete. And again, if we can stretch our time sense out and sit on these places for long enough to get the long-term perspective on what's going on, that we will benefit from them and come to understand these communities better. And the last thing is I keep thinking I'm asking the questions about how these things work in the wrong way. I'm asking the wrong question. And I keep thinking, if I knew a little bit more, or I worked a little bit harder, or I was actually a lot smarter, I could figure it out and figure out a way to pose the question in such a way that the path through this complexity to understanding how these communities work would be self-evident and obvious. And I just haven't been able to do that. So as I sort of slip into decrepitude and senility here, I leave that challenge to all the young'uns of you that are out there and that are interested in these kinds of problems to figure it out. What's the question that we need to actually ask that will lead us to the answers of understanding of these complex and truly fascinating sorts of biological communities? Okay, so acknowledgments, of course. I really have to acknowledge all the summer students that have submitted themselves to the summer class and learned the things that I've tried to teach them, and then returned that by helping collect a lot of these data. Especially all of these folks here who are very special and have helped me in a lot of different ways across the decades here. All my stalwart dive buddies who are way too numerous to mention across all the decades. And then lastly, every one of the people here who've taught me anything about the fascinating sort of biology of Monterey Bay. That has enriched my experience here and helped me both as a scientist and a teacher. So I thank you all, and I'd be happy to answer questions. Questions? Chuck.
I have a question. You've been doing this for 40-odd years, and you started off with 3 species that you really found out a hell of a lot about, and you didn't have anything to say about them in the kelp bed over the rest of 40 years, except they showed up a few times in starfish stomachs.
Yeah, they get eaten a lot. They get eaten a lot by sea stars.
So are they one of the uninteresting things that just didn't show any dynamics?
Actually, there hasn't been much going on with the turban snails.
To me, that's one of
Yes.
the really fascinating things.
Yeah.
Because people always look at things that change.
Right.
But things that don't change are also really interesting.
No, the turban snails, a part of it is they're so abundant, it's really hard to get good numbers on their populations. They're hard to sample that way. But they're not the only gastropod that stayed more or less constant. A lot of the work that John and Theo did on the abalone out here, and then we've taken it over in the summer class too, in that 40 by 40 meter area around the 100 meter mark, the abalone population, the red abalone populations there have been incredibly constant over the last 40 years. And so even though a lot of the stuff I talked about had to do with changing things but on differing timescales, there have been things that have been just incredibly constant. There's one place where I was chasing turban snails around out in the kelp bed here. It has a little rock ledge with a big anemone sitting on it. And though I haven't gone back every month and shook hands with it, there's still an anemone on that same rock wall that I was working on back in the late 70s. And as far as I know, it's the same individual.
We had some anemones that showed up in Rafe's quadrats that we thought may have been the same individual that were counted
by Hewitt. Yeah. Yeah.
But just an observation that I'd really like to thank you as an invertebrate biologist, that you pointed out that Piesaster was really smarter than sea otters.
collectively.
Because they adapt their diet very regularly with abundance of food and its availability.
David.
I want to take Chuck's comment and amplify that. That you brought up that we shouldn't look at otters at least as automatons. Probably all animals. So otters are mammals, so they presumably can learn and think. And humans are taking care of abalone and sea urchin populations because they like them. They learn that they're good to eat. And maybe even, as you said, maybe even the invertebrates are learning also.
Oh, the invertebrates definitely learn. They're a lot smarter than we think. They continually amaze me in their ability to do what they do. And if you ask the questions of them in the right way, they tell you some fascinating stories. But you have to ask them the right way and then listen to the answer rather than assuming what the answer is going to be.
Well, let's assume that in a month we get a fruity otter out there. And it starts the process of eating sea urchins.
Yeah.
How quickly do you think the sea urchins could be mowed down?
Once my prediction, and it's probably not worth very much, but my guess would be once the otters figure it out and really start eating urchins, it will probably be less than a year before the kelp starts growing back in patches, and a couple years before it'd be back to kind of where we wouldn't recognize that a deforestation had even occurred. Because the same thing happened in Carmel Bay on a localized deforestation event out there that Chris Harold and I looked at back in the mid-'80s. And within a couple of years of the reduction in urchin populations, the whole community, the sessile community as well as the owl community, was pretty much back to normal.
Thank you, Jim. I was blown away by the talk. I was wondering, when you're describing all these dynamics about barnacles, sea urchins, sea stars, and it kind of struck me, will there be some more non-consumptive, interaction that might have to contribute to the patterns that you're describing over a long period of time, rather than actual predation per se, but actual chemical interactions, how urchins behave differently, and how they forage differently in the presence or absence of starfish, as asked earlier. Do you think that might have played some role in that dynamic? There have been some ideas about not only the consumption of urchins by sea stars like Pycnopodia, but just the presence, because the urchins have a pretty good escape response to it, that it changes their behavior, their grazing behavior, and indirectly affects grazing intensity. So certainly those kinds of indirect sorts of effects are important. On sort of other kinds of organisms, interactions for space. Space is really limited out there, so a lot of the sessile stuff, which I really didn't talk much about except for the barnacles, a lot of the sessile organisms are interacting for space, and so there's lots of those kinds of stories that didn't get a chance to be told today too. So yeah, it's not just predator prey, certainly, and hopefully I didn't give the impression that that's all that's important out there, because there's lots of other stuff going on.
So oftentimes for measuring success for restoration, we use baselines to compare. And in the future, how do you think Hopkins is going to be useful, given these years, decades of data, to help us really measure restoration offshore in the intertidal to the future?
Yeah, I think the sort of real difficult part of the question is sort of what we mean by restoration. And I think if it were up to humans to actually put a kelp forest back together, I don't think we could do it. I think the best that we can do, given the complexity and the dynamics of these communities, is to give nature a chance to do it itself by easing up on whatever stresses we've inflicted on the environment or those communities. And then I always like to think, even in my darkest days when I despair about the negative effects our species is having on our world here, is that biology is incredibly resilient. And it always finds a way somehow. You know, you look, and then being an invertebrate zoologist and thinking about evolutionary patterns over long periods of time, you think about the Permian extinction, when 90-some-odd% of all the coastal species went extinct, and yet we have great diversity of things today, 250 million years later. And so that's all due to biology and the resilience and ability of biology to make do and take what each individual organism and species does to make it from one day to the next. And the aggregate of all that put together results in this huge pageant through time of diversification and success of biology. And so I think despite the fact that what we can do to our environment and damage to some of the habitats out there, that it's transient. And when we're gone, the world will recover, and it will be something different than it might have been if we had stayed present, but biology will find a way. And I think trying to restore things is it's important that I think we concentrate on giving nature a chance to restore these communities, because at least I'm not wise enough to know to what state to restore a community that needs to be restored. But I think nature is. So we need to give them a chance to do that.
How long do you think we've had a macrocystis kelp at Hopkins Marine Station?
Over what time spans? You know, because McLean in his paper in 1962 describes out here being mostly abalone. This is just prior to the otters moving back into southern Monterey Bay. And in the late 50s, early 60s, he describes it as mostly bear, urchins, abalone, big abalone, and a few bull kelp and other things.
Because macrocystis has been moving northward.
Yeah.
You know, I wonder what its introduction here, is the dominant algal species making a kelp forest really dates back to.
Yeah, my suspicion is that before we started harvesting otters for their pelts, there was probably at least intermittent kelp forests here.
You don't think it was Nereocystis? Because Nereocystis was very abundant early on, documented off here. We don't have it now.
Actually, there are some clusters of Nereocystis that are showing up out this way in the barren areas where the otters have removed the macrocystis. So I don't think I'm wise enough to predict what things were in the past. I have a hard enough time remembering the things I've experienced directly, let alone a past that I haven't seen. But that's an interesting question.
Well, yeah, because that is a baseline kind of question.
Right.
Maybe we better let Jim go, restore his memory. He'll be around till the end of the summer, so you can always ask questions later on.
Thanks.